I am a realistic optimist. Because of the time constraint, I'm going to read a lot of this, but I feel deeply about it. I believe that we can improve the quality of life of those on the autism spectrum in the coming years. By quality of life, I mean greater independence through improved communication, better impulse control, reduced repetitive thoughts and behaviors, and enhanced intellectual function. Each generation talks about breakthroughs, but speaking from years of experience, I sense that something is different today. And I'm very much influenced by technological breakthroughs. Genetic variants mutations have been identified that enhance the risk of autism. Advances in gene sequencing have made it possible to search the entire human genome with high accuracy in less than 1 week at a cost of about $1,000. The first human genome sequence, published 15 years ago, took a consortium of large laboratories several years to complete at a cost of $1 billion. This million-fold reduction in cost, along with revolutionary advances in analytic tools, now allow us to rapidly investigate thousands of individuals. And given the heterogeneity of autism, the autism spectrum, and the rarity of individual mutations, this will be necessary to define the full autism genetic landscape. There have been remarkable advances in our abilities to manipulate genes, possibly leading to gene therapy and correcting mutations. Advances in neuroscience I will get to in a few minutes. There's been a remarkable influx of talented investigators, as Allison just described, in recent years. Scientists from all areas of biology, from mathematics, physics, engineering, and chemistry, have turned their attention to the nervous system and to autism. I like to think that the Simons Foundation played a big role in this transformation. Our annual meeting, organized by Louis Reichert, John Spiro, and the entire science staff, just was completed 2 days ago. They're all seated here on the left. It was an intense, rewarding experience. Attendees claim that it is the very best meeting that they ever attend, and they come away with the feeling that the time is right now to develop new hypotheses and seek new collaboration. We know that there's an increased burden of mutations in people on the autism spectrum compared to their unaffected siblings and unrelated controls. In 2003, there was one autism risk factor in which we had a great deal of confidence. Today, we have uncovered 71 new mutations that enhance autism risk. There are new, that is, de novo mutations in the germ cells of one parent or the other. Most of them are killers in that they destroy the protein that they encode. It is now clear that other types of mutations occur that damage but do not actually destroy the proteins, but they cause smaller but significant effects. Altogether, several hundred genes may be involved. No need to throw up our hands in bewilderment and frustration. Far from it. Statistical analyses have identified gene networks that converge on certain functions that may very well reveal previously unsuspected targets for therapeutic intervention. One point of convergence involves synapses, the point of contact and communication between cells. There are 100 billion neurons in our brains. That's recently been downgraded to 80 billion, perhaps because of the election year debates. But the neurons are transparent, but they can be stained and visualized under appropriate illumination. The blue neuron on the left is covered with hundreds, perhaps thousands of dots that are synapses that have arrived from nearby and distant input neurons. In the cartoon on the right, a synapse is shown in cross-section. It contains many vesicles that are packed with chemical transmitters that on cue are discharged into the cleft, separating the communicating cells. Two transmitters are indicated on the right. GABA, gamma-aminobutyric acid, the primary inhibitory transmitter in the brain, and glutamate, the principal excitatory transmitter. One important hypothesis states that autism results from an imbalance between excitation and inhibition. There seems to be an imbalance either caused by excessive excitatory drive or reduced inhibitory drive. My bet is on the reduced inhibitory drive and the defects in GABA because numerous genetic and physiologic studies point to a deficit in GABA synthesis and action in the brain. Clinical trials are underway to investigate drugs that may augment the action of GABA. How do these, and what is next? How do these molecular machines called synapses work? Aside from the synthesis and release of transmitters, there's a complex network of proteins and genes that determine the maturation and stability of synapses and their ability to change with experience. Of course, these processes must be investigated. We must determine which changes in synapses are involved and when they occur during development. Current studies point to prenatal, before birth changes in key synapses, probably involved in the cerebral cortex. There are obviously other changes, but that's a good starting point. An important question that must be answered concerns how do these synapses affect neural circuits? And ultimately, how do such neural circuits control behavior and thought? The possibility of insight into the function of the social brain, which is, after all, responsible for our evolution as a species, it what drew many of us to neuroscience in the first place. All animals, worms, fish, flies, mice, rats, monkeys, and humans, are welcome in this study. This is a picture of a zebrafish viewed from above. At this age, the fish is transparent. The cells have been labeled with a gene that encodes a protein that fluoresces, glows green when the cells are active. It's a new way to monitor the activity of thousands of neurons simultaneously. Can you play that video? Now, the fish is a social animal, and when a stimulus is played that indicates that she has fallen behind her neighbors, she gets active. First, she thinks about where am I, and then in very short time, lights up the neurons in the back of the brain that trigger movement and in which direction and where to go. This is a very recent video which promises great results in the future. Now, I have another video to show you, which is a little bit unusual for this audience, but it is a film of deep brain stimulation of an individual with Parkinson's disease, if you could play that, that has a lot of lessons for autism.
By the time we met Sybil this past spring, even a simple task like eating breakfast was a frustrating battle with her own body. How can you live your life when you're shaking so much?
It's extremely difficult, sometimes even overwhelming. And I get very emotional. Sometimes I cry. It's just a hard thing to do.
The disease had gotten so bad that sometimes her muscles froze completely, making her face almost expressionless. and her legs almost useless. The woman who once was always on the go could barely move, confined to a wheelchair. But as the disease got worse, the drugs did less.
One of the things that this disease has really accomplished is it has really robbed me of my dignity, my self-esteem, because it has taken away my independence. When you have to be totally dependent on other people to do everything for you, you actually lose your self-respect. You feel worthless. And that's what Parkinson's has done for me.
But on the morning of the surgery, Sybil was confident, even as nurses fitted her with a head frame to help guide surgeons during the operation. Deep brain stimulation comes down to one thing: location. Finding the target area holds the key to Sybil's future. The device electronically records the activity of individual cells within her brain, so the doctors can actually hear the trouble spots. That noise is the sound of a neuron misfiring.
Can you move your ankle up and down? It's hard to do for her.
Finally, the doctors think they have placed the electrode in the exact location that will help Sybil. When they turn on the stimulator, she suddenly shows an astonishing range of motion in her legs and hands.
Pick a leg up at all? There you go. Up and down. Drop it down now. Now pick it up. Great. And she couldn't do that before. So. How about the hand? Can you open and close your hand for me now? Good.
She was doing so well that Dr. Vitek had a surprising suggestion: more surgery. He thought that a second implant in the right side of Sybil's brain could get rid of the tremor in her left hand and might improve her stride.
The minute they got in and he hit the target, it's like magic.
Good. Okay, open and close your hand.
He said to me, open your hand and close your hand. And so I knew right away. It's amazing.
Sybil now had two stimulators, one for each side of her body.
How do you feel?
Great.
What can you do now that you couldn't do before?
Oh, good heavens, look at that. Look at her. I feel as if I've been given my life back. And I'm taking hold of it.
So I met Sybil. I do get choked up every time I watch this. I met Sybil 15 years ago and showed this video to a congressional subcommittee, and that resulted in a $1 billion 5-year plan to attack Parkinson's disease. And we need that in autism research. Is this a cure? No. The degeneration of nerve cells in the substantia nigra in her brain is not fixed. But is it effective? Yes, in that symptoms are reversed and her quality of life is dramatically improved. Alison Singer, I remember a quote from the Charlie Rose show, which we appeared together, in the most dramatic moment of that show, when she said, "I wish I could get inside of my daughter's head for one moment and tell her that I love her." So this is as close as I've seen that we can come. DBS is extended beyond Parkinson's disease to various neuropsychiatric disorders, including OCD, depression, severe anxiety, and epilepsy. And the thought is that some form of stimulation, either through implanted electrodes or extracranially, may be in order for autism. But what are the targets? Where are we going to attack? Well, there's a good roadmap building for autism in the brain. One is involved in the medial prefrontal cortex and the cingulate gyrus, and on the outside of the brain, in the superior temporal sulcus and the temporoparietal junction. Are these targets that might be attacked by stimulation? And most of all, can we use everything that we learn in the coming years to attack risk factors medically and surgically and develop more effective therapies? This will take a huge national effort. It's not just, it's be spearheaded by the science, but it will take people in law, business, arts, and the humanities to help reduce the stigma associated with autism and provide the resources to build community resources like that created by Greg and Laurie Ireland in Florida that will bring people along and welcome them and make them satisfied in their lives while we wait for these revolutionary changes. Thanks.